Just before we break for lunch, we've now got 15 minutes to ask any questions that we may have to our speakers. So I'd just like to invite you all up to the stage, please.
So I might start off with a question for Ellie, which is about the artwork that you had in your presentation, which was amazing. And what's the source of that?
Yeah, so I create the artwork for pages. Yeah, I'm cartoonist for the Phage Journal as well. So.
We did have a question. Was it hand-drawn or was it AI?
Oh, no, it's hand-drawn, yeah.
Thought so.
Sorry? Oh yes, you can buy them online. I've got a shop on Redbubble if anyone needs anything with phages on them.
I have a question for Jack. On the core culture experiment that you did, you mentioned that you did it for 24 hours. Would the outcome would have been different if you would have done like 5 cycles after every 24 hours with the same core culture? And second question, you mentioned you picked the filtrate of each of the core culture experiments. And did you determine if the bacterial sequences of the PA01 had prophages in them and the metabolites as well? What were the metabolites from the core culture?
So for the second question, we haven't had a look at analysing the co-culture yet. That is to be done later on in the PhD. Could you just clarify, sorry, what you mean by 5 cycles?
So when you did the core culture experiment, you were only doing it for 24 hours. I think I got that from your slide.
Yeah.
So if you do the first 24 hours, yes, assuming that is the first cycle, continue incubating it, take an aliquot from the first cycle, introduce into the next cycle with the same like 10 microliters up to the 7, maybe 5 cycles of it. Would that outcome be different, like you're looking at the evolutionary or the fitness across the same, but I don't know if I'm making some sense.
No, I think that makes sense. So
yeah.
For the time culture, we actually did 20 hours and 24 hours in preliminary trials, and just the data was kind of allowed for enough growth at 24 hours, which was why that time point was chosen. I'm currently in the progress of a co-culture assay where it is transferring it daily into new culture. I did a trial one of that for 10 days, and in AMI R1, which was one that both in live assays and co-culture assays caused a reduction in fitness. It did seem to almost die out in terms of growth, Pseudomonas strain, and then recovered after about day 3, but I need to analyse that a bit further.
All right, thank you for that, because we are also doing almost the same thing with core cultures, and we observe the dying after 2nd or 3rd cycles of different bacterial species. Thank you.
Thanks.
And I had a question for Chris. I was really interested in the mutually exclusive pathways bit you were talking about, and the different hypothetical mechanisms you spoke about all sound really interesting. I was wondering whether you or any of your collaborators are doing any work testing or looking into those mechanisms and how you're going about doing it.
Well, no, is the answer. We wish we could. These are the things that we need to investigate. The big caveat is this is all computer stuff, and we need to go into the lab and we need to do this. So the things I outlined are the things that I would love to do next.
Thank you to everyone for your talks. Great variety of interesting presentations. So I have questions for Eleanor and after that for Chris. Eleanor, first of all, fantastic artwork. And the question I had for you is, have you tested these phage cocktails that you're developing in polymicrobial systems?
It's a good question. I'd like to, but we haven't tested them in proper polymicrobial systems. So when we're testing the white swarms, they obviously have their own microbiomes in there, and we can see that there's other bacteria in there, but we haven't been able to build out these polymicrobial systems yet. The funding for the Klebsiella side isn't there currently, so obviously I'd love that, and if somebody had a big pot of money, then yes, we'd love to build up these polymicrobial systems.
Yeah, it's always boils down to money, unfortunately. Would be interesting to see the results. Yeah, and the question for Chris, I actually have two questions. So first of all, about the machine learning part, Lucy and you trained this machine learning algorithm based on the coding sequences, right? Have you looked at the non-coding sequences or some structural elements of the genome? Would this potentially improve the machine learning predictions?
So the first one, what we did, yes, the answer, first of all, is the presence and absence of protein coding genes is basically the machine, the engine underneath it. But we did look at using a tool by Nick here in the room, which tries to identify possible missed genes or other elements. Again, they would be protein coding genes if they were missed, whether it would increase the predictive power. And in that case, it didn't. So so in terms of the complement of protein coding genes that we have, we seem to be at the limit of that. Yeah, the next question would be whether or not we could enhance that with other types of genes like regulatory elements. Currently, the approaches that we were utilizing, like pangenome approaches, are not well suited to that type of kind of functional elements. But again, that's an area that's, I think, in development.
Thanks. And second question about the gene compatibility and incompatibility pairs. Have you seen any enrichment for any specific functional categories? For example, for genes, you gave an example of the two transposases are not compatible in Pseudomonas aeruginosa, as far as I remember. Are there any specific categories where you see these incompatibility pairs, or it's just like randomly distributed?
Yeah, it's an area that we still don't know enough about, because quite often they're not the canonical AMR genes. They are, we predict them to be important to the expression of the AMR phenotype, but they are these accessory genes. And as I said, sometimes they're efflux pumps, and sometimes they're genes with just, they have a very ill-defined function, but they seem to be important. So it's another area of where more work is needed.
Great, thank you.
We have one more question down here.
Thank you. To Chris, really, can you explain why some species, some strains are MDR and very rapidly and emerges MDR, whereas others seem, and I don't know if it's part of this genomic architecture that you spoke about, that almost from the introduction of antibiotics are still antibiotic gene free.
Okay, the first part, I can't answer it specifically, but I can give you my hypothesis. In terms of rapid MDR, it seems that, like, if you have resistance in some cases to one particular antibiotic, you're halfway there to being resistant to another, because we can see that certain types of genes are shared, are necessary for phenotypic expression. They might be, for instance, an efflux pump might be a very common thing that multiple resistance mechanisms require for a phenotype. So that's one possible explanation. When you have one set of genetic architecture, you're already predisposed very quickly to being able to do that. That's a possibility. Sorry, what was the second part of the question again?
Why some never seem to host AMR genes?
Yeah, that's a question I'm not sure I can answer. I do think that there's a question about the genetic, as we've been calling it in our paper, genetic grammar. Like, in some cases, for sure there are some sets of genes that an organism can have, which means that certain type of resistances are just not possible, because they have a gene which is so important to their metabolism or life cycle that they are not going to lose that, but it conflicts fundamentally with the genes required to express a phenotypic resistance. So there might be a case, so that's again, it's a hypothesis, but a case where it's the genetic architecture of the organism itself somehow contains things which are fundamentally excluding the ability of some of acquiring certain types of resistance. So
I can't think of an example, but I'll go away and think of one.
It is difficult, but these are where we were speculating. But it could be as simple as a shared regulatory element that is, very broadly affects the expression of multiple genes, for instance. Let's imagine a situation like that. You have this regulatory element, but it is perhaps, conflicting with the expression machinery for the resistance genes, for instance. So you could have a case where you have regulatory conflicts in a cell, or various things like this. And again, it's speculation, but these are the questions that we would love to try and follow up and try to figure out.
Hi, question for Chris. Did you consider looking at mutations when you were building these models? Because they could obviously change the function of genes and be a source of potential discordance with genotype and phenotype.
Yeah, it's a good question. We went out of our way to make sure we didn't include any genes like that, because we couldn't, at the scale of the genome data sets we were dealing with in thousands of genes, we couldn't guarantee we would accurately identify whether certain specific mutations were present or not. So we went through the approach of only looking at those type of resistance mechanisms where it was the presence or absence of a gene that conferred that was associated with the resistance, and we avoided any of those mutation-specific resistances.
Hi, I've got a question for Ellie. Ellie, when you're screening your phages, how often are you finding genes that increase the fitness of the bacteria, and what genes are common?
So we haven't really found any virulence genes in those phages. So because we're concentrating on lytic phages, so those that kill their host rather than integrating into the genomes, we avoid any lysogenic phages because they're more likely to carry antimicrobial resistance or virulence genes. So we haven't so far in the lytic phages found any, but we just have to keep screening those, and we've worked with groups at a phage centre in Finland, and they also do their own screening of those before they get used on any patients too.
Sorry, just a quick question to Ellie about phages and their suitability for the next stage, and what would you, in a sentence, feel that the UK needs to do to get them into some sort of clinical stage?
Yeah, so so far in the UK, they've only been used in named patient trials against diabetic foot ulcers or on specific compassionate usage. Yeah, I think one of the struggles is that they're classified as a medicine and they're regulated to the same standard as molecular medicines, so all produce the same GMP standards, where they have to be biological identical. And these are very stringent, but because it's an evolving biological entity, it's maybe not appropriate. So if we were able to introduce the same system that are used in the Belgium system, where you have a whole raft of phages which are done to near GMP standards and ready to mix and use off the shelf, because currently in the UK we can use phages from outside the UK if they're at whatever regulatory level they need to be in that country, but we can't produce them in country to that level and use them in the UK. So it's a real blockage that we have to go outside the UK to get phages that we can use inside the UK. So it's a lot of it is going back to Belgium, getting those phages. And there's people at the Becky Meyer Centre at Leicester who are trying to build up this UK phage bank, and there's people like Josh Jones, who's at UK HSA, who's trying to set up better links between clinicians and the people with the phages now. So there is more momentum to get past these obstacles, speaking to MHRA about how to regulate them and what might happen, and DEFRA as well of what happens to them in the environment, what we can use. So it's a lot of regulatory hurdles, a lot of red tape, and lack of understanding by all these different people. So it's trying to get the information out there, trying to share what we know and what different countries have found useful.
Do you think there's a role for the public to understand and then put pressure?
So I have seen a lot more interest in phages recently. So there is some kind of grassroots understanding that phage is useful, but I think it's trying to balance it currently between it's not the next homeopathy or alternative medicine versus what are the scientific facts behind it. And so there is more momentum. There are people who are asking now for phage therapy. There is a bit more understanding, and it's trying to keep that in line with using it as a medicine. So you can buy over the internet, you can buy probiotics which have phages in them, but the ones that we've tested in the lab can't actually isolate phages out of them, so don't know if they contain any live viruses. So yeah, there is understanding and there is momentum in the public. It's just kind of connecting it all up to make sure it's based in science and facts.
Thank you. We have a question at the back. Oh.
I know I'm loud, but I have to use the mic for the people online. So I have a question for Lauren. So you were seeing some reduced evolvability or even survival in your respiratory mimicking medium. So what is the implication for that? Because I'm guessing your respiratory mimicking medium is more representative of the infection in a respiratory tract, right? So I know at the minute you're only looking at 2 organisms at the same time, and perhaps maybe you have more than 2 organisms at the same time when you have a respiratory infection. But what is the implication for that? Are we overestimating? Are you saying that we are overestimating resistance whenever we're using TSB? And perhaps are you saying that in clinic, maybe that resistance is not as bad as it seems from the fact that the organisms are not actually surviving properly in respiratory mimicking medium?
Yeah, I think it's hard to make a full conclusion on it because in a infection environment, there's going to be so many different factors that do affect how bacteria evolve resistance. But I've seen in this that maybe there needs to be more looked at. Like, we can understand fundamental mechanisms of resistance evolvability within lab nutrient-rich media, but maybe for the actual sense of inside someone's body in an infection, that we need to actually look at more factors that may influence that and how that might affect mechanisms in the evolvability too, that can then go back to like other things. So for going forward, I'm now looking at species interaction within this evolution, so that's then going to play a factor. So there's so many different things within the infection niche that might cause different evolution than what we're seeing in lab media. So I think it needs to be considered.
Okay, do we maybe have any last questions from the audience or online? No, okay. Thank you. I want to thank again our speakers for this session. And we're going to be breaking for lunch until 2:05 with a poster session starting at 1:15. And we're going to reconvene for an interactive session. So get refreshed.
Thank you.